Samson.
Yep.
Hi, I'm Melissa Runge-Morris, and I am the director of the Institute of Environmental Health Sciences at Wayne State University. It's my pleasure today to introduce our guest seminar speaker for the Integrative Environmental Health Sciences Seminar Series hosted by IEHS, CURES, and CLEAR. You know, Dr. Samson Jamesdaniel is one of the star faculty members of IEHS. He first joined our team of faculty in 2014. And the reason why we asked him to speak with us today is because of his exemplary role as a researcher and the very, very interesting work that he does, which is focused on hearing health and the role of the environment as a modifier of the quality of our hearing. Dr. Samson Jamesdaniel is what every academic researcher aspires to be. That is, he does molecular mechanistic work, trying to understand at the molecular level how environmental and pharmaceutical agents affect the quality of our hearing. So he uses animal and cellular models to do that research, but he has also been successful in taking his research to humans and has done research involving first responders and people who are vulnerable to environmental exposures in the field and also at risk for detrimental effects on their hearing quality. That's part and parcel of their occupational health risk. Dr. Jamesdaniel was educated in India. He received an MD degree in alternative medicine at Tamil Nadu MGR Medical University in Chennai, India. He also received a PhD degree in physiology from the University of Madras, Chennai, India. Interestingly, he took his research to the postdoctoral arena and actually did a very interesting postdoc in Lodz, Poland, where he was at the NOFAR, I think, Institute of Occupational Medicine, where he studied the dynamics of hearing and events that impair our quality of hearing as we proceed in our life. He also did a postdoctoral research associate experience at the Center for Hearing and Deafness at the State University of New York in Buffalo. Dr. Jamesdaniel is funded by NIH. He's a principal investigator, and he has also had DoD, Department of Defense funding, because as you can imagine, military personnel are exposed to environmental toxicants in the field, and among other stressors that affect their quality of health, they have effects on their hearing and sense of well-being related to that. So without further ado, I'd like to give Dr. Samson Jamesdaniel a chance to give us his seminar today. The title is Ototoxic Interactions of Environmental Stressors. Thank you so much for joining us today, Samson.
Thanks, Melissa, for the nice introduction. And I'd also like to thank the organizers for giving me this opportunity to share this research. So in this talk, I'd like to talk about the ototoxic interactions of environmental stressors, particularly a physical stressor, which is noise, and another chemical stressor, which is going to be lead. Let me just, okay. So I'd like to begin with sharing a few stats about hearing loss. More than 1.5 billion people, nearly 20% of the global population, live with hearing loss. And among this population, almost 430 million, that is 1/3 of this population, have disabling hearing loss. And in the United States, Hearing loss is the third most common chronic physical health conditions after hypertension and diabetes. And hearing loss affects the quality of life and productivity. And if you consider first responders like firefighters and also the military personnel, this can have serious consequences. And hearing loss is also linked to cognitive decline, dementia, Alzheimer's disease, and depression. And it is noted that reduced cognition and dementia increases almost 5 times with the risk of reduced cognition and dementia increases 5 times with severe hearing loss. It also has devastating effects on children because it affects their linguistic development, education, and social integration. And it also imposes a huge economic burden. If you consider the veterans' benefit claims, almost more than 32% of the benefits are being claimed for hearing-related disorders. So this is a major public health issue. And as you see in this chart, the world's population is projected to grow, and it is likely to be as high as 9.7 billion by 2050. And it is also estimated that almost 68% of this population is going to be living in urban areas. So, and the World Health Organization also projects that the prevalence of hearing loss is going to double during this period. So it is probably many of the factors, the ototraumatic agents that are in the urban environment are likely to play a major role in this dramatic increase in the prevalence of hearing loss. So our research is mainly focused on understanding what are these agents, how do they cause hearing loss, how do they interact, and can we find any strategies to prevent this increase in the prevalence of hearing loss. So our lab mainly focuses on 2 different stressors, as I said. So one is noise, as you can see here in the map on the left side. So this is Detroit, as you can see. The Metro Detroit area has higher levels of noise when compared to the adjacent regions. Similarly, if you look at this map on the right side, so this is a map of the percentage of housing built before 1980. As you can see, a large number of houses were built before 1980, which means they are likely to have lead paints because lead paints were banned only in 1978. So we have this kind of a lot of exposure to noise as well as hearing, as well as lead. And our study is focused on understanding the interaction between noise and lead, because the noise-induced hearing loss is well characterized and many studies have been published. However, lead-induced hearing loss is not that much fully characterized. There are studies that indicate it causes hearing loss. There are other studies that state that it might not be that ototoxic in adults. So we wanted to thoroughly investigate the effect of lead exposure and noise exposure, as well as their interaction on auditory function. So there are several different targets for ototoxicants, and as you can see in this illustrations, so this is the cochlea, which is the peripheral auditory organ. So it is usually coiled, and here for descriptive purposes, it is shown in an uncoiled form. So this is known as the organ of Corte, where you have the sensory receptor cells, that is the hair cells. And auditory signals are transmitted through these ossicles. So you have the stapes, and it passes through, and it stimulates the hair cells. And in this cross-section, as you see, these are the hair cells. Once the hair cells are stimulated, they send these signals down through this auditory nerve, which goes through different pathways through the auditory brainstem and finally reaches the auditory cortex. And the illustration here shows that in the apical region, the sounds that are low frequency sounds are perceived, whereas in the basal region, high frequency sounds are perceived. So this is also important because different types of sounds affect different regions of the cochlea. And more about these cochlear targets. The reason that I'm talking about this is the data that you will see when I show from our lab. So we are mainly looking at hair cells, how hair cells are affected by these exposures. So these are the 3 outer hair cells, and we have an inner hair cell. And then we are also trying to see the effects of these exposures on the ribbon synapses. So you have this kind of the ribbon synapses, which transmit the signals received by these hair cells. So from this inner hair cell, so the signals are transmitted through these synaptic fibers, and they reach the spiral ganglion. And from this spiral ganglion, which is the neuronal cell, the signal is further transmitted through the auditory nerve. So we always look at the effect of these exposures on the hair cells, the ribbon synapses, and the spiral ganglions, which are considered as major targets for many ototoxic exposures. So you'll be seeing a lot of pictures about these structures. So moving on to lead-induced hearing loss, as we all know, lead is ubiquitous. It is found in paints, batteries, pipes, and it is predominantly absorbed by inhalation and ingestion. And its neurotoxic effects are well characterized, but we also know that lead exposure disrupts the structure as well as the function of the auditory system. However, the ototoxic potential is not fully understood because there are studies that show that short-term exposure to lead doesn't cause significant hearing loss in adults. So that is what we are trying to find out, what is the dose-response relationship and how it causes hearing loss in these after these exposures. So this is the schematic of the study design for us here. So we have these 5-week-old CBHA mice. We chose this specific model because these are young adult mice models that we expected to be susceptible to lead exposure because we wanted to understand the mechanism by which lead causes hearing loss. So we chose this age group. And we exposed them to a pretty high level because we wanted to understand what are the targets in the cochlea and how lead affects these different organs. So we used 2 millimolar lead acetate in drinking water for 28 days. So that increased the blood level to almost 400 nanogram per ml. So this is pretty much a high level lead poisoning levels in mammals. So, but this level of lead exposure did not induce significant loss of weight. And then after exposing these animals, we tested their cochlear structures. So we used immunohistochemistry to look at different regions in the cochlea. And then we also did a proteomic analysis, mass spectrometry analysis to look at the cochlear synaptic proteins. So initially, we looked at the outer hair cell activity. So we can assess this using a test known as distortion product ototoxic emissions. So when we tested this ototoxic emissions with different frequencies, so we tested the 8 kilohertz, 16 kilohertz, and 32 kilohertz, we did not see any change after lead exposure. So the amplitudes of these distortion product emissions were pretty much similar to the controls in the lead-exposed animals. So this indicated that the lead is not affecting the activity of the outer hair cells. And then by immunohistochemistry, we looked at the outer hair cells because these outer hair cells are highly susceptible to many toxicants and also to noise. However, with lead exposure, we didn't see any loss of outer hair cells in both basal region, middle region, and apical region. So we then now moved on to look at the auditory brainstem responses. However, we see a 5 to 10 dB shift in the hearing thresholds, indicating there is some damage, but it's not happening in the hair cells, but in some other regions. And it was across multiple different frequencies. So 16, 24, 32, and 4 kilohertz, we have significant shift in the hearing thresholds. So we wanted to look at other regions that might be potentially damaged by this lead exposure, as we knew that outer hair cells are not being affected by this level of lead exposure. So we looked at the ribbon synapses. So we used 2 different biomarkers. One is CTBP, which is a presynaptic biomarker, and GLU-A2 is a postsynaptic biomarker. So these synaptic biomarkers, we looked at their expression levels in the synapses, which are attached to the inner hair cells. And as you can see, lead induces a significant decrease in the levels of both these synaptic biomarkers. So this indicated probably lead is targeting the cochlear ribbon synapses and causing cochlear synaptopathy. We also confirmed this using physiological measurements. So when we do the ABR measurement, we can actually quantify the wave 1 amplitude, which is an indicator of cochlear synaptic function. As you can see, that there is a significant decrease in the wave 1 amplitude with click stimuli and also with 8, 24, and 32 kilohertz frequencies. So this indicated that lead exposure is actually affecting the ribbon synapses. And we also looked at the pairing of these synapses, which is also, again, an indicator of how the synapse function. As we see here, there is a significant decrease in the number of paired synapses after lead exposure. So all of these results indicate that lead is probably targeting the cochlear ribbon synapses rather than the hair cells. So we actually wanted to look at the changes in the synaptosomal proteins, cochlear synaptosomal proteins, because lead is targeting the ribbon synapses. And we actually detected almost 4,947 proteins. Many of them were significantly increased and some of them were decreased. And then when we did the bioinformatic analysis after this, we found that the synaptic vesicle cycle pathway is highly enriched. So this is again another confirmation that lead is targeting the synaptic vesicle. So now we wanted to understand what is the pathological mechanism underlying these toxic effects. We know that lead actually causes neuronal damage by inducing excitotoxicity, increase in calcium influx, and also causing oxidative stress. So we wanted to look at the oxidative stress changes in the cochlea as well. So we looked at different changes in the different genes related to oxidative stress using a PCR array. And we found that many of them were significantly changed by lead exposure. And in other studies, in our ototoxic, in the drug-induced ototoxic studies, we found that when there is oxidative stress, it also causes nitrative stress, and nitrative stress has been a significant or critical factor in inducing ototoxicity. So we wanted to check whether lead exposure causes nitrative stress. And mainly we wanted to see in the spiral ganglions, because the lead is causing the toxic effects in the neuronal segment of this cochlea. So when we looked at the spiral ganglion cells, we see a significant increase in the nitrotriazine levels after lead exposure. So it was, significantly higher after lead exposure. So this indicated that lead is actually inducing nitrative stress, and it is actually targeting the synaptic and the synaptic region and the spiral ganglion cells. So this nitration, as I said, we found to be a critical factor in cisplatin-induced ototoxicity. And this is actually caused by the interaction of free radicals, that is nitric oxide radical reacts with superoxide radical to form peroxynitrate. And this peroxynitrate leads to the formation of different one-electron oxidants that oxidize the tyrosine molecule in the protein. So not all proteins are nitrated. There are certain proteins that are susceptible to nitration, and we found in the literature that many of the synaptic proteins are also susceptible to nitration. And this nitration can have significant consequences because it can alter protein function, it can cause proteasomal degradation, and it can also modulate phosphorylation cascades. So we found from this study on lead exposure in animal models, we found that synaptopathy is a major outcome, and probably nitrative stress is driving this toxic outcome. So now we wanted to see whether noise can also induce a similar effect, because noise is known to affect the hair cells, but we want to see whether nitrative stress is a critical factor in noise-induced hearing loss as well. So our noise exposure model is illustrated over here. As you can see, we are exposing the animals to 90 decibels of noise for 2 hours per day for 14 days. And then we are testing them after 28 days. The reason that we are not doing the testing immediately is we wanted to assess the permanent threshold shift. If we assess these threshold shifts immediately, there'll be a temporary shift, which means it'll show increased hearing loss, but that is going to come down. So we wanted to assess the permanent effect of this noise exposure. And as you can see here, noise significantly increases the thresholds shifts after noise exposure, almost in all frequencies and also after click. And unlike lead, what we see here is noise also increases the distortion product amplitude. So it affects the outer hair cell activity as well, whereas lead did not affect it. And we can see that in the immunohistochemistry images over here with the significant loss of hair cells, outer hair cells in the basal region of the cochlea. So noise actually targets the outer hair cells. So now we wanted to see whether it also affects the spiral ganglions and other regions. So, and we also looked at whether noise induces nitrative stress. As you can see here, there is a significant increase in the nitrotyrosine levels in the noise-exposed cochlea, particularly the basal region, and it has been quantified. There is a significant increase. And when we looked at the spiral ganglions, as you can see here, it also increases the nitrative stress. And not only the spiral ganglions, protein nitration levels are increased, but it also decreased the spiral ganglion density. So when the spiral ganglion density is decreased, it can affect the transmission of auditory signals because adequate amount of spiral ganglions are required to transmit the signals properly. So when the density is decreased, it can affect the transmission of auditory signals. So we find that noise also induces nitrative stress in the outer hair cells as well as in the spiral ganglions, and it affects their function. And when we looked at the ribbon synapses, as you can see here, so we are looking at, again, the CTBP2, which is a presynaptic biomarker, and GluR2, which is a postsynaptic marker. And we are also looking at the pairing. So with the controls, you see both of them, the presynaptic and the postsynaptic markers are paired, which means the signal is transmitted, whereas in the lead-exposed specimen here, shown here, and sorry, the noise-exposed specimen shown here, you see a gap in between, which means they are not fully paired. So again, similar to what we saw with the lead, in the noise exposure also, we see a decrease in these, in the punctal levels of these biomarkers. And also the pairing of these synaptic biomarkers is also decreased after noise exposure. So it seems like noise and lead both are targeting the synapses as well as the spiral ganglions. And noise, in addition, targets the hair cells as well. And here is again a confirmation looking at the physiological measurements. So the Bay1 amplitude is significantly decreased here in the basal region after noise exposure, and the latency. So the amplitude actually increased the number of neurons that are firing, and the latency indicates the time taken for firing, and the time taken is also increased after noise exposure. So this indicates that noise induces cochlear synaptopathy. And when we looked at the nitration of proteins after noise exposure, we see number of proteins are nitrated. And again, as in the lead exposure, we see synaptic vesicle cycle as coming as one of the highly enriched pathways. So this indicates that noise probably is targeting the synaptic vesicle or protecting the synaptic vesicle may be a good strategy for preventing the ototoxic effects of these types of exposures. So now we wanted to look at the combined exposure. So we know that noise actually targets synapses, spiral ganglions, and the hair cells, and lead actually targets the synapses and the spiral ganglions, and both of them induce nitrative stress. And so we wanted to see how a combined exposure is going to behave. So in this model, what we do is we do this 14-day noise exposure. And in addition, we also simultaneously do this 28-day lead exposure. And we do the testing, hearing testing after the 28-day exposure. And the blue trace here indicates the control. So the hearing thresholds are pretty much normal over here, whereas the lead indicates there's a shift compared to the controls. Noise also indicates a greater shift at some frequencies, whereas when you have this combined exposure, it has a significantly higher levels of hearing threshold shifts when compared to isolated exposures. And one of the things that we found that was it seems to have an additive effect at some frequencies and synergistic effect at some other frequencies. And also when we looked at these amplitudes, so these are the ABR waves, and when we measure these amplitudes, this also indicated probably it is causing synaptopathy. And immunohistochemistry, when we did the immunohistochemistry analysis, it indicated that noise this combined exposure actually induces nitrative stress in the hair cells, as well as the spiral ganglions. So this is consistent with what we saw with those isolated exposures. We see significant involvement of nitrative stress in driving these outcomes. So what we wanted to do is we wanted to see if can we target this nitrative stress to prevent these toxic effects. So we used a metalloporphyrin MNTBAP. So this is a scavenger of peroxynitrate. So it can scavenge peroxynitrate. And as I showed in the illustration before, peroxynitrate is found by this interaction between superoxide and nitric oxide, and that leads to the nitration of tyrosine in the proteins, resulting in protein nitration. And when we scavenge this peroxynitrate, it can decrease the nitration of protein and also inhibit the nitrative stress. So we used this MNTBAP, metalloporphyrin. And what we found was, as you can see here, so these are 4 different groups. We have the controls. And this is the MNTBAP alone. We have this lead plus noise, so which is significantly higher, the threshold shift. And when we did the MNTBAP treatment, it brings it back significantly. So this was happening in almost all frequencies, so indicating a kind of an autoprotective effect of MNTBAP, suggesting that inhibition of nitrative stress or targeting nitrative stress could be a good strategy for preventing the ototoxic effects of these environmental stressors. And again, similarly, we also see in the distortion product autoacoustic emissions, we see a significant protective effect with this lead, noise, and MNTBAP treatment. So when compared to this green trace, as you see here, lead and noise, it significantly protects in different frequencies, suggesting the autoprotective effect of targeting nitrative stress. And as we also see with the wave and amplitude, which kind of indicates whether there is cochlear synaptopathy. So again, here, MNTBAP treatment significantly protected. As you see here, it actually protected cochlear synaptopathy from happening due to these exposures. And it was observed in all different frequencies. So these data suggest that nitrative stress is probably oxidative stress, and nitrative stress is probably a critical factor, critical mechanism that is driving the ototoxic effects of lead and noise exposure, and targeting this nitrative stress using these pharmacological compounds could be an promising strategy for preventing these ototoxic outcomes. So in addition to evaluating these underlying mechanisms, we also wanted to investigate the auditory health outcomes in a vulnerable population. So what we did was we entered into a collaboration with the Detroit Fire Department here. So because the firefighters in Detroit are exposed to generally higher levels of noise because of the noise generated by these machineries that they use while fighting fire, and also from these trucks. And as I showed you in the map earlier, that Detroit has a very old housing stock. And therefore, when these firefighters go and fight the fires in these stations, they are exposed to lots of these lead dust. And most of the time they have this protective gear on them. But what we, from our interactions with the firefighters, what we found was after fighting the fire, when they go back in to see if there are any remnants of fire, they usually go without these protective gear. And at that time, they are likely to be exposed to all of these dust particles that are suspended in the air. So we wanted to see whether these firefighters are a good population to study this interaction between noise and lead, because they are exposed to these kind of agents regularly in the line of duty. So our study design was using different surveys. So we used 3 different surveys. One was to assess the hearing handicap inventory for adults. So this was used to assess if they have any difficulty in auditory perception. And then we used another survey, tinnitus functional index, to assess whether they have tinnitus. Tinnitus is a kind of a perception of a phantom sound when there is no external auditory stimuli. So that is a kind of an indicator of auditory damage. So when the hearing is affected, so some can experience this tinnitus. And then we also had another survey in which we were trying to assess the history of other exposures, because whether they were taking any other ototoxic drugs, whether they are exposed to recreational or other environmental noise, and those kind of factors, and how often they are using their personal protective equipment, and information related to prior exposure. So we used 3 different surveys. We assessed their lead levels. So we initially used the urinary lead level assessment. And then later on, we switched to toenail assessment, because toenail is a kind of indicator of a little bit longer term exposure to lead rather than urinary levels. Then we used audiograms to see how their hearing function is. So this is kind of a very early data that we got. So what I just want to point out here is you see this notch here in the 4 to 6 kilohertz frequency region. So this is kind of a typical notch that you will find with noise-induced hearing loss. So when a patient has a noise-induced hearing loss, they will usually have a typical notch in this 4 to 6 kilohertz region, which is known as a speech frequency. And we saw this notch in a number of firefighters. So these are some of the audiograms that you see here illustrated over here, and you see here number of firefighters, even in this limited sample, had this typical notch. And in the early data, we also found that some of the firefighters had tinnitus based upon the survey that we conducted. But this is again a data of, I think this includes almost 111 firefighters here. And as you can see here, 21% of these firefighters had slight degree of hearing loss, 20% moderate, and another 10% had severe hearing loss. So when you compare this to the statistics that I told earlier, which is 20% of the adult population, general population have hearing loss. Here, when we combine all of these things, it comes to 51%. So it indicates that these firefighters have a higher degree of hearing loss when compared to the general population, and probably that is due to these occupational exposures to many of these ototraumatic agents. And the assessment, the assessment of hearing handicap indicates that 29% had mild to moderate hearing handicap, and 7% had significant handicap. So overall 36% of these firefighters report some degree of hearing handicap. And when we combine these together to analyze these together, what you see here is the pure tone average of 4 and 6 kilohertz. And here is the HHIA, hearing handicap score. And what you see here is these are the firefighters who have a higher hearing handicap score, but their hearing thresholds are pretty good. The PTA, the pure tone average, is pretty good. So this is suggesting probably hidden hearing loss. We can do other tests to assess the hidden hearing loss, but this is kind of an indicator, preliminary indicator. Probably some of these firefighters are not able to clearly perceive the auditory signals. So this is also very important because these firefighters have to have really good hearing when they fight or when they look for rescue, when they are in research, the rescue and search operations. So they need their hearing to be good. But this indicates that almost 13 firefighters from this population have some degree of hidden hearing loss. And then when we did the toenail analysis, so we find that of the 26 firefighters we did initially, we had 9 of them having lead levels that were higher than the median concentration found in welders who are high risk, which is in a high risk operation for lead exposure. So it seems like firefighters are having significantly higher lead levels, even greater than welders, suggesting that probably they are having some higher levels of lead exposure in their occupation. And then this is, again, we analyzed the audiograms after dividing the firefighters based upon their lead levels into 4 quartiles. And as you can see, so this is the 4 and 6 kilohertz frequency region, and the hearing levels are hearing thresholds are much higher for the higher quartile, suggesting a potential association between lead levels and hearing threshold shifts, whereas we don't see that kind of a shift over here. So these data suggest that probably lead and noise are contributing to the higher levels of hearing loss that we observe here in our firefighters, in our Detroit firefighters. But again, I would like to note some of the limitations of this study. This is kind of a cross-sectional study. We are just looking at one term. If you do a longitudinal study, starting to do these measurements before the firefighters join duty and then following up after 3 years or 5 years, we would have a better understanding of these association between these exposures and hearing loss. So with this, I would like to summarize. So in summary, our studies indicate that outer hair cells are not the primary targets in lead-induced ototoxicity. However, lead exposure causes cochlear synaptopathy, and oxidative and nitrative stress may be critical factors in resulting in these lead-induced ototoxicity. Lead also alters the abundance of cochlear synaptosomal proteins, and concurrent exposure with noise exacerbates the lead-induced hearing loss. And we found that targeting a shared signaling pathway like the nitrative stress prevents the hearing loss induced by concurrent exposures. And firefighters are probably vulnerable to hearing loss induced by noise and lead exposure. So what we want to do in our future studies is we want to further assess what are the other age groups that are susceptible to lead-induced hearing loss. So we would like to see whether perinatal exposure is causing hearing loss. We already did with young adult exposure. We also want to compare that with adult exposure. And the other thing that we would like to do is we have started with a very high level of lead exposure. So we wanted to bring it down to multiple low levels and see whether a level that doesn't cause hearing loss during isolated exposure causes hearing loss when we have this combined exposure with noise. We also want to bring the noise levels to a little bit lower level, similar to traffic noise, and see whether these interactions are manifested when we have these combined levels of exposure that doesn't cause hearing loss when they are isolated exposure, but cause hearing loss when we have combined exposures. So these are some of the studies, and I would also like to note we are also working with other exposures, particularly PFAS, in collaboration with Mike Petrola's lab, and we find significant changes in the hearing function and also morphological changes in the cochlea. So we would be interested in looking at whether firefighters also have these kind of PFAS exposures. So these are some of the ongoing studies, and I would like to end here and acknowledge the current and past lab members who contributed to these studies, and also our collaborators and funding agencies. And I'd be happy to answer any questions that you may have at this time. Thank you for your attention.
Thank you very much, Samson. Excellent presentation. Lynn, you might be able to see the attendees a little bit better than I can to see if anyone has any questions for Dr. Jamesdaniel.
We just
no.
we got one from Carrie just now. If you'd like me to read it out loud, Dr. Carrie Leach, who is part of CURES, she, let's see, says, I know the CURES CAB has members of the firefighting community back in 2014, given that the CAB, which is the Community Advisory Board, didn't have a way for you to connect with this hard to engage community. How did you go about gathering data and leaders? Do you have lessons learned to share with researchers who are engaging hard to reach populations?
Yeah, it is a struggle to reach to these kind of populations, and it is difficult. Initially, yeah, we worked with some of our collaborators who had connections with the fire department, so that went well. After you reach a critical amount of firefighters, it is difficult to go beyond that. For example, we reached 111 firefighters, but our target was around 200 initially. So it is difficult, and the number of firefighters available is also limited because we just had a focus on just the Detroit Fire Department. We didn't want to go to other fire departments. So I think we were targeting almost 1/4 of the total number of firefighters in the department. So that was a little bit difficult. So yeah, if we had other sources that would help with this recruitment process, that would be helpful. Yeah.
Graham, I see your hand is up.
Yeah, I want to first of all thank Samson for that excellent presentation, and just to show I've been around long enough to remember when Samson gave an excellent community presentation in a church basement, and it really struck me, without wishing to be ironic, how it meant so much to the community to be heard about such a profound effect on their quality of life, to know that people at Wayne State were doing research on hearing loss, which is something that, you know, you don't really see big headlines talking about that. I have a couple of questions. One is to do with, I think in the general population, it's fair to say that hearing loss is associated with people feeling lonely, feeling isolated, feeling depressed. I guess I kind of wanted to flip that. You were doing surveys. These are firefighters that have an ego. Is it almost like a badge of honor to be losing your hearing in that kind of profession? Do you want to comment on that, Samson?
Yeah, I wouldn't say that, because even some of the firefighters who had significant shift in their pure tone average, when we looked at their surveys, they were answering, okay, we are hearing pretty good, but the audiograms indicate that they have hearing loss. So some of them don't even realize they have that degree of hearing loss. So I don't think that they consider it as a badge of honor, because it may also affect their career prospects, because if they are found to be having significant degree of hearing loss, they may not be able to continue to work. So I don't think that is an issue there. Yeah.
Good point.
Yeah.
Samson, I had a question. Hearing loss with the fire department, OSHA requires that if you are exposed to over, I believe, 85 dBAs, that you should have a hearing loss program. Do they have a hearing loss program where they regularly monitor and take measures of hearing loss within the fire department?
Yeah, as far as I know, I think they have to monitor their hearing, but I'm not sure at what intervals they have to do. And it is if they have certain degree of hearing loss, I think they may have to report, and they have to take measures to address that issue. Okay. But I have not seen a kind of a structured program in which they have to do it at specific intervals. But I may be missing, because I'm not fully familiar with all their programs that they have.
Okay.
Graham, you have a question?
Yeah, I think it's beyond impressive that you've actually not only characterizing a problem, but you're also suggesting possible interventions that could help either mitigate or even restore the loss, which especially with hearing. I mean, I'm thinking of work of like people like, and I hope I get his name right, Cheng Hu here at Wayne State, that's developing stem cell models to try and replace hearing. I guess I'd be interested to know your opinion on whether you think such interventions are likely to bear fruit, or do we have to concentrate on your route of correctly characterizing the nature of the loss to try and come up with an intervention?
Yeah, as far as the interventions are concerned, we are mainly focused on prevention rather than restoration, because as far as the field is concerned right now, these hair cells, if they are lost, they cannot be recovered. Once they are lost forever. So it depends upon how much degree of hearing loss you get because of the loss of hair cells. On the other hand, if the damage is in other regions, particularly like what I showed with the lead exposure, if it is in the cochlear synapses, there are studies those can be restored. So it depends upon where the damage is and where the toxic effects are happening. And as far as the stem cells and the regeneration studies are concerned, I think people are still trying to see if they could regenerate the hair cells, but it has not come to fruition. They are still trying to find out if the hair cells can be regenerated. But as of now, prevention is the best strategy because restoration is not ideal once you lose the hair cells.
Thank you.
Samson, if I could ask a question. So you showed really nicely that the nitration of the cochlea cells in the cochlea was pretty extensive, really.
Yeah.
Is there a surrogate marker that could be used that I imagine it's pretty hard to take samples of cochlea from people. So can we look at a blood biomarker, for example, or something else that might give us a clue that somebody should be concerned and should be looking for ways to decrease their exposure or increase their use of something that would counterbalance that process?
Yeah, I haven't seen any biochemical or biological markers because they are relying on these physiological measurements with these here audiograms and hearing thresholds. Because as far as nitrative stress is concerned, I think cochlea is a very small organ, and to have that come through the blood and have a blood test to be developed or some other measures that could be developed, I haven't seen those kind of things so far. And people mostly rely on these physiological measurements to assess how much damage is being caused. I haven't seen. And there are some other tests that people are trying to, but I don't think that they are in the markets or they are not fully developed yet.
Okay, thank you.
Yeah.
I don't see any more hands up. I don't know about you, Lynn, but oh yeah, you have your own hand up, Lynn.
Yeah,
right.
I have a question that I think I'll put out there. I happen to know quite a few people on the Detroit Fire Department. And one thing that seems to be pretty popular among those folks that I know on the department is attending very loud, heavy metal kind of rock concerts and that type of thing. So is that taken into consideration? Is there a control for that type of outside of occupational hazards type of hearing loss?
Yeah, so that is a very good question, and that is something that we are also struggling. And every time you submit a proposal, that is something that I get a comment. How do you disentangle the effects of noise versus other exposures in these firefighters? So it is a difficult thing. So What we are doing right now is just using these surveys in which we are asking about all of these types of exposures, whether they have this recreational exposure, environmental exposure, occupational exposure, do they go for hunting, those kind of things. So we are using that kind of an assessment that may not be an exact measurement of the amount of loud noise that they are exposed to, but we get some kind of a sense about how much noise they are getting exposed, or the practices that they are doing. And yeah, so that is all that we are doing right now, but that is a issue for us to actually assess how much noise they are getting, other than the occupational exposures. In terms of occupational exposure, there are some measurements depending upon the role they play there. If they are a driver or something, so some other roles, they have some kind of measurements that are standard measurements over there. But outside of their occupation, it is difficult to assess. And we have to rely on these surveys to determine how much exposure they get.
One more thing.
Yeah.
So in the department, there are dual role firefighters who do both fire and EMS work. And for a very long time on the department, they were completely separated by EMS and firefighting.
Yeah.
So I was wondering if there has been any difference noticed between those two groups. I believe that those on EMS are constantly in the ambulance, hearing the sirens pretty much all day long. And firefighters, of course, they work very hard too, but maybe they're not in the fire truck as much as the EMS folks are in the ambulance. So is that making any kind of noticeable difference?
Yeah, unfortunately, I'm unable to comment on that because we have a very small sample size. And our sample size is also mainly focused on firefighters. So if you look at the EMS people, I think we have very few, because our target population was mainly firefighters, and the sample size is also too small to kind of glean those kind of differences.
That makes sense. Thank you. I was just curious.
Yeah, in future studies, if we go with these kind of longitudinal study and also have another case control group in which we are actually having people who are not exposed to these kind of exposures, I think we'll have a better answer for these questions. Steve, yeah.
So sorry I showed up late. I was excited for this one and I got triple booked, so I'm so sorry. I don't know if it had come up yet, but I'm thinking of the personal air quality monitors that they're strapping on folks. Is there a way to...
Steve, sorry to interrupt you, but I think you're away from your microphone. It's very difficult to hear what you say.
Right, this silly microphone, and it's giving me problems these days. Is this better?
No, but I can sort of hear you. We could try to repeat what you say.
My guess is probably you were asking if just like the air quality monitors, do we have anything else for measuring the noise exposure? Is that your question?
Yes, on person level, like a personal monitor.
Yeah, so we have these dosimeters and people have used that to study the level of exposure, noise exposure, but they have used that in training conditions, but in actual live fire, when they fight the fires, I don't think that they have done a study with these kind of dosimeters. So this actually applies for just the noise exposure. Yeah, I think it may be a difficult thing for them to actually put on the monitors when they are in a rush to go and out for fires. But people have studied that, putting those dosimeters on them in training conditions.
If you used it for for the non-occupational exposure and had some folks wear some outside of their job duties, you might get some more measurements to basically address the non-occupational exposures. And rather than putting it on person, if you could put it on the rigs and have a monitor on each rig, would probably be helpful for the occupational if you get a chance.
Yeah, that's a good idea. Yeah. But in terms of long-term exposure, I think we might still find it difficult to kind of have a cumulative exposure of these noise levels that they are exposed to.
Thanks so much. Appreciate it.
Thank you.
I see another one in the chat from Carrie Leach once again. Have the firefighters you have connected with asked for information or recommendations based on your findings?
We haven't shared this data yet with the firefighters. We are still working on this. And one thing that we were planning was probably we will give a talk without identifying those things for probably a group of firefighters. And then once we complete this toenail analysis, we were planning to give individual firefighters who were interested in knowing about their results. So we haven't initiated that conversation yet.
Okay, I don't see any more. Oh, Carrie says we can help with that, Samson.
That'd be
Consider
great.
me
Yeah.
a resource. So thank you,
Yeah,
Carrie.
that'd
It's
be great.
wonderful.
Yeah.
I don't see any more questions in the chat. So Dr. Franklin just got in there with, is the union interested in your finding?
Yes, the union is one who gave us the very good supportive letters, and they were the one who interestingly participated in many of our outreach events. So they are really interested. But in our previous study, we had kind of a very limited recruitment or criteria for recruitment. So that actually eliminated some of them. We cannot have older firefighters who are above 55 because they will have age-related hearing loss. We wanted to have firefighters only after 5 years service because we wanted to have occupational exposure. So that limited the number of firefighters. But the union is really interested, and both the president, secretary, they gave a very good letter even for our grant applications that we used. So they are really interested.
Okay, have we got any more questions for Dr. Jamesdaniel today? We're at 1:27 p.m., so we're just about done here, I think, unless somebody has something they need to ask. But at this point, if I could just talk about our upcoming seminar series speakers. Let's see, I'm sharing my screen now to show who we have coming up. We have a gap next week, but on the 6th of March, we have Dr. Shanique Brown joining us from Baruch College, and she's going to be discussing strategies for effective team science. So please look out for these upcoming events. You can see at the top here, we have all these events.
We don't see your whatever you're sharing there.
Oh, that's unfortunate. I'm sorry. Is that better?
No, we're looking at your email or something. Oh, there you go.
No, I saw a list of speech talk.
Saw it very briefly, but then it went away.
I saw the same thing.
Yeah, I saw the same. I saw the list.
Okay.
Okay.
How's this? Is it back?
Great.
Hoping so. Okay, so there it is. If you would like to join us, you can always find the event links here, events.wayne.edu/research-events. Those are all listed there. We keep that list up to date, or you can reach out to me, the project coordinator for CLEAR and CURES. My name's Lynn, by the way. Hello.
And also someone, Carrie, did you ask whether these are virtual going forward? They are for now. There is some repair work and upgrading being done to the seminar room in iBio. So we had so many technical difficulties with that work in progress that we shifted to Zoom so that we could have these things without a lot of technical business interrupting the process. So when we get further word on the seminar room, we'll let you guys know. All right, that's all I know. Goodbye, everybody.
Thank you for joining us today. And thank you
you.
to Dr. Jamesdaniel. That was a good one. I enjoyed it.
Thank you.
Bye, everybody.